Welcome to Chapter 17, Blood, Part 1. This is Part 1 of 2 lectures for Chapter 17. In this part, we will have a brief introduction on what blood is, as well as its functions. From there, we will move on to talk about blood plasma, and then we will spend the rest of the lecture talking about erythrocytes, or red blood cells. We will talk about the production of them, their function, as well as their fate and destruction. In Chapter 1, we introduced 11 organ systems found in the human body. One of those organ systems was the cardiovascular system. Remember, an organ system includes different organs or structures that come together to serve common functions. Blood belongs to the cardiovascular system, in addition to the heart and blood vessels. Blood is really a transport vehicle that we will see many substances use to move throughout the body. Several of the functions of blood are listed here, and we will take the next few minutes to go into detail with each of them. Blood functions as, again, a transport system. It regulates certain processes in the body, and it also serves to protect. Blood is the major means that substances like respiratory gases, nutrients, and even metabolic wastes will use to move throughout the body to go from tissue to tissue or an organ to another organ. Blood will transport oxygen and nutrients to working muscle and working tissue. Also, it's going to remove carbon dioxide and metabolic wastes and carry them to organs like the kidneys and the lungs to be expelled or excreted. And lastly, blood will pick up hormones from endocrine organs and deliver them to their target tissue or to their target organ. In addition to transportation, blood also helps to regulate certain processes or factors in the human body. The first thing that blood does is helps to maintain body temperature, and this is either for hot or cold. Blood is responsible for absorbing but also distributing that heat throughout the body, and also carrying blood to the surface of the body to dissipate that heat. Blood also helps to maintain a normal pH level. It does so by acting as a reserve for alkaline or basic ions, such as bicarbonate. And lastly, blood is going to help maintain fluid volume, and it does so with different types of proteins that we will see that are found in the bloodstream. Lastly, blood serves to protect our body several different ways. The first is by preventing blood loss. We will see that there is a constituent of blood known as a platelet, which will help to seal broken blood vessels, therefore preventing the loss of blood. And lastly, in the blood, we will have certain types of cells known as leukocytes, or white blood cells, that are responsible for preventing infection or providing immunity. In Chapter 4, we learned that blood is a type of connective tissue, and we usually don't think of it as a tissue because it's a fluid, and it's the only fluid tissue in the human body. By definition, a tissue includes cells with similar structures that come together to serve a common function. And remember, any type of connective tissue includes 3 elements: fibers, ground substance, which both make up the extracellular matrix, and cells. When we look at blood as a tissue, the extracellular matrix is now called plasma, or blood plasma. This is the stuff that surrounds cells, or formed elements. We have 3 formed elements in blood: erythrocytes, which are also known as red blood cells; leukocytes, which are also known as white blood cells; and platelets. Blood is an example of a suspension when looking at mixtures, and that is when it looks heterogeneous, meaning you can tell the difference between the solvent and the solute. Now, if you take a test tube of blood and stick it in a centrifuge or a device that spins it around really fast, it separates these components out from each other. And when you do that, the test tube now has 3 layers. The bottom layer is entirely made up of erythrocytes. Now, here's a term in bold that you absolutely have to know: hematocrit. The term hematocrit refers to the percent of blood volume that is made up of erythrocytes or red blood cells. Now, you don't have to know percentages for male and female, but you do have to know the definition. Sitting on top of the erythrocytes, we now have a buffy coat, which is an off-white layer, primarily made up of white blood cells and platelets, or the other 2 formed elements of blood. And sitting on top, taking up the majority of the space, is the plasma, or the extracellular matrix. If you're having trouble picturing what I just covered, here's an image for you. Again, you take a test tube of blood, seen towards the left near number 1. You stick that test tube in a centrifuge, which is a device that spins the tube around rapidly to separate out its components. And when you do that, now your test tube of blood shows 3 layers. Again, the bottom layer is made up of erythrocytes, or red blood cells. They settle to the bottom because they are the most dense. Sitting on top of the erythrocytes, we now have a very thin layer known as the buffy coat, which includes the other 2 formed elements of blood, leukocytes and platelets. And to top it off, we have the blood plasma, which is the extracellular matrix of blood as a connective tissue. We will start by focusing on the extracellular matrix of blood, known as the blood plasma. And this is the part of blood that is responsible for transport. We don't really have nutrients and hormones and wastes found in red or white blood cells. They are actually dissolved in the plasma. We find nutrients, we will find some gases, hormones, wastes, and proteins. The majority of the proteins are going to consist of albumin. You have to know the functions of albumin. Albumin is going to be there to serve as a carrier for other molecules. It'll help serve as a blood buffer. Remember, a buffer is an organ or a system that can function either as an acid or a base. It's there to resist abrupt swings in the pH. Albumin will also contribute to osmotic pressure or the movement of water across a blood vessel. Now remember, a tissue or a connective tissue includes the extracellular matrix, which we now know is plasma, and formed elements. We have 3 formed elements in blood: red blood cells, white blood cells, and platelets. However, out of those 3 elements, only the white blood cells are considered a complete cell. That means that red blood cells and platelets lack a nucleus, as well as other organelles. And we will see that later on when we focus on erythrocytes and platelets. Platelets are simply cell fragments, or little bits and pieces of what was once a larger cell. And we will see that blood cells, remember, come from red bone marrow found in the skeletal system. Remember, one of the functions of the skeletal system was hematopoiesis, or the formation of blood cells. On this slide here, we have a microscopic view as well as a blood smear seen under a microscope of blood tissue. And from this view, we are able to see different blood cells like red blood cells, which are the red disc-looking structures, or the red dots. You can see white blood cells, which are the yellow-colored balls in the top image, or they have some blue stain to them in the bottom picture. And then we also see a little bit of platelets in there. Again, those are cell fragments, so they will just look like tiny bits and pieces. For the remainder of this lecture, we will focus on one of the formed elements in blood known as the erythrocytes, or a red blood cell. Overall, we will see that red blood cells are these very small diameter cells, and they're basically just bags of oxygen and carbon dioxide. Their main function is to transport respiratory gases. Remember, erythrocytes are not a typical cell. They are anucleated, which means they don't have a nucleus, and they also lack organelles. So it's basically just an empty bag. But it's filled with what we now know as hemoglobin. We saw myoglobin in muscle tissue. Hemoglobin is a molecule that will bind oxygen and allow for red blood cells to transport gases. We really can't talk about structure and function separately without mentioning the other. Now, the red blood cells are a great example of the complementarity of structure and function. So take their structure for example. They are a bag filled with hemoglobin, which binds oxygen. There's no space taken up by a nucleus. There's no space taken up by organelles. So we have all of this area and surface area and space inside of the cell to fill with hemoglobin. Approximately 97% of erythrocytes are filled with hemoglobin. Again, red blood cells lack organelles like mitochondria. We know that mitochondria use oxygen to produce ATP. If a red blood cell had mitochondria, by the time that red blood cell got to a working tissue, the mitochondria would have used up all of the oxygen already. Let's talk a little bit more about hemoglobin. Remember, hemoglobin, the term heme means blood. Globin means that it binds oxygen. The majority of a red blood cell is comprised with hemoglobin. That is how we are able to transport respiratory gases using red blood cells. Don't worry too much about the anatomy of the hemoglobin molecule, but what you should know is that one hemoglobin molecule can transport 4 oxygen molecules. Within that hemoglobin molecule, we have some polypeptide chains, but we also have an iron atom. We have 4 chains, and in the center of each chain, we have iron. Each iron binds 1 oxygen, which allows for a total of 4 oxygen molecules per hemoglobin molecule. Here we have 1 hemoglobin molecule. There are 4 chains. We have 2 beta chains and 2 alpha chains. In the center of each chain is an iron atom. Iron also gives blood its red appearance. So looking at that red disc, that represents 1 iron. Each iron will bind 1 oxygen. So if a hemoglobin molecule has 4 irons, it can bind 4 oxygens. Now that we have a brief understanding of the structure of a red blood cell as well as its function, let's talk about where they come from. Remember that one of the functions of the skeletal system was hematopoiesis, or the formation of all blood cells. That occurs in red bone marrow. We find red bone marrow in the epiphyses of long bones, such as the humerus and the femur. We find red bone marrow in the girdles, specifically the coxal bones or the hip bones, as well as in some vertebrae. All blood cells, all formed elements, red blood cells, white blood cells, and platelets, will come from a hematopoietic stem cell, which you may also see known as a hemocytoblast. Again, this is a stem cell that can give rise to red blood cells, white blood cells, and platelets. Now, from there, we will have certain factors like hormones and growth substances that will tell that hemocytoblast what direction to go in. A new term that you may not be familiar with is now called erythropoiesis. If you see that prefix, erythro, you should think red blood cell. Hematopoiesis was the formation of all blood cells. Erythropoiesis is the formation of red blood cells. There are 8 transformations or 8 phases that these cells go through prior to actually becoming a red blood cell. Again, all formed elements come from a hematopoietic stem cell. From this list, you simply have to know the order in which this blood cell transforms. You do not have to know about that stage or the descriptions given to you here. So after the hemocytoblast, we have a myeloid stem cell, and we will see other blood cells come from the myeloid line. After that, we have a proerythroblast, which transforms into a basophilic erythroblast, a polychromatic erythroblast, orthochromatic erythroblast, a reticulocyte, and then we have finally our mature erythrocyte. However, if you look at an individual's reticulocyte level or numbers, that is a pretty good indicator of their red blood cell count. You will not see this image on your exam, but it again lists out the different transformations. Again, all blood cells come from a hemocytoblast, and it originally has a nucleus and organelles. But once it makes that decision to go and become a red blood cell, we see the organelles disintegrate and we see the nucleus die off. So eventually we end up with an erythrocyte, which is a bag filled with hemoglobin that functions to transport oxygen. While we always have a steady production rate of red blood cells, there are certain factors that will increase production or even inhibit production. If we have too few red blood cells, that leads to tissue hypoxia. Hypoxia is a decrease in oxygen. Too many red blood cells, now everything's too crowded. That's going to increase blood viscosity, which is the thickness of blood. And we have more than 2 million red blood cells being produced per second. Again, that's typically our basal rate, but there are certain factors that can stimulate or inhibit production. And we will see one hormone in particular that will play a role in production. And we will also address some dietary requirements that affect red blood cell production as well. The one hormone that you have to know is known as erythropoietin. Shortened, it is EPO. Again, there's that prefix erythro, you should automatically think red blood cells. We have a constant rate of EPO in our bloodstream to maintain that basal rate. However, in response to factors like tissue hypoxia, we will see an increase in release of EPO by the kidneys. The kidneys pick up on low oxygen levels and stimulate the release of EPO. EPO will communicate with red bone marrow and increase the production of red blood cells. You may be familiar with the Olympics or the Tour de France, but sometimes these athletes will use an external source of erythropoietin. It's often referred to as blood doping. What happens when they take an external source of EPO is they have an even higher rate of red blood cell production. And they do that to increase their performance or enhance their endurance, because the more red blood cells they have, the more oxygen they can provide to themselves. Now, There are certain causes of tissue hypoxia or a decrease in oxygen levels. You have to know these three causes. One cause of low oxygen levels would be due to a hemorrhage or maybe an increased rate of red blood cell destruction, so decreased red blood cell numbers. Another example or cause of tissue hypoxia is an insufficient hemoglobin per red blood cell. This could be caused by an iron deficiency. Oftentimes, people with iron deficiencies might feel lightheaded or weak, and that's because they cannot carry enough oxygen. And lastly, if you are in a place or location with a reduced availability of oxygen, such as a high altitude, or even someone who has a lung issue like pneumonia, that will lead to tissue hypoxia. All of those causes will communicate or stimulate the kidneys to release EPO. Now, there are certain factors that inhibit EPO and slow down red blood cell production, such as too many erythrocytes or high oxygen levels. Here we have this teeter-totter again when we look at homeostasis. Right now, our homeostatic value or factor is normal blood oxygen levels. Now, if those blood oxygen levels drop, it's due to a decreased red blood cell count, maybe caused by a hemorrhage, a decreased amount of hemoglobin, often seen with iron deficiencies, or a decreased availability of oxygen. Those three factors that cause tissue hypoxia will stimulate the kidneys to release EPO. EPO heads to red bone marrow and increases red blood cell production. The more red blood cells we have, the greater our ability to carry oxygen. So we bring those blood oxygen levels back up to a homeostatic level. In addition to erythropoietin, there are also certain substances or molecules that we intake through our diet that are required to regulate or stimulate erythropoiesis. We find amino acids in protein sources, lipids and fat, as well as carbohydrates. But something else that is required for erythropoiesis and red blood cells is iron. Remember, we saw iron or 4 irons in each hemoglobin. Without iron, we cannot bind oxygen. But iron does not float freely in the bloodstream because it's toxic. If iron is going to be stored in a cell, it will bind with ferritin or hemosiderin. But as soon as that iron is called upon and has to move throughout the body or be transported in the blood, we now bind it to transferrin to transfer it. But in addition to these macromolecules and iron, vitamin B12 and folic acid are also extremely necessary because these cells rapidly divide, like developing red blood cells. Now, maybe you have assumed this, but red blood cells don't live forever. They live for approximately 100 to 120 days. And again, that is from the time that they become a mature red blood cell until they begin to break down. Again, red blood cells do not have a nucleus. They are incapable of growing and even dividing. So eventually, red blood cells become worn down, and we see them end up getting trapped in the spleen. If you're unfamiliar with the spleen, it is found in the left upper quadrant in the abdominal cavity. When you think about breaking down a red blood cell, here we have a nice schematic with everything that happens to the 3 parts of a red blood cell as we break them down. Again, erythropoietin, produced and released by the kidneys, travels to red bone marrow to stimulate the production. But after about 100 to 120 days, these red blood cells get trapped in the spleen. And from there, we see the three parts, the heme, the iron, and the globin, break down. And they each have their own way in which they are destructed or broken down. Make sure you know what happens to each part. This was chapter 17, part 1. We introduced blood as a tissue. We identified its three formed elements. We learned about plasma. And then we finished up with red blood cells, or erythrocytes.